The overall goal of the following experiment is to assay gene expression using functional nuclear machineries extracted from cells. This is achieved by first harvesting and swelling the HeLa cells. Next, the cells are lysed by dowsing and then centrifuged to pellet the nuclei. In the third step, the nuclei are salt extracted, concentrated, and dialyzed in order to obtain soluble and functional gene expression machineries. Ultimately, the functionality of the nuclear extracts can be evaluated based on their transcription and splicing activities, as assessed with a coupled RNA polymerase II transcription and splicing system.
The main advantage of this technique over existing methods like bulk nuclear extract preparation is that with this technique, a small amount of cells can be used to prepare the nuclear extract.
Begin by plating HeLa cells on 3 150 mm plates with DMEM media supplemented with FBS and antibiotics. Grow the cultures in a 37 degrees Celsius incubator with 5% CO2 until the cells reach 90% confluency. At this point, aspirate the media from the HeLa cell cultures and wash them once with 13 milliliters of room temperature PBS per plate. Then aspirate the PBS, stand each plate on its side for a few seconds, and aspirate the rest of the PBS. Next, use a cell lifter to scrape the cells to the bottom edge of each plate, and then pool the cells in a 1.5 milliliter Eppendorf tube. Finally, pellet the cells in a microfuge for 5 minutes at 100 times G and at 4 degrees Celsius. Now aspirate the